Hello friends, welcome to Microbiology Insights. In today's session, we are going to discuss about how to observe a microorganism. So as we all know that microorganisms are so small that they cannot be observed through naked eye. So in order to observe a microorganism, there are two steps. One is the preparatory step and the other is the observation step. So in the first step, we need to prepare a microbe so that they can be observed under microscope, and in the observation step, we observe that particular prepared microbe through microscope. So the question arises that why we need to prepare a microbe to observe it under a microscope. So microbes, basically they are translucent in nature as a cell, means their refractive index is more or less similar to the surrounding. So that's why they cannot be observed through a microscope. So we need to color this microbe so that a contrast is created between the microbial cells and the background, and we can observe it under an optical device. So the preparatory step, it involves several sub-steps. So what are they? So basically, the preparatory step involves smear preparation, and staining, while the observation step involves use of microscope to observe the stained microbe. So these are the two steps with the help of which we can observe a microbe under a microscope. So the first step of observing a microbe is the preparatory step, which involves smear preparation and staining. So in order to observe a microbe, we first need to culture a microbe. So microbial culture is microbe grown in artificial media in laboratory condition. So such culture is the prerequisite for observation of a microbe. So we need a glass slide first, and we take a loop full of culture using a microbial loop. Like say this is a microbial culture in a conical flask. So we use a microbiological loop prepared out of nichrome wire, which looks like this. Okay, so we dip this loop in this culture and take a loop full of culture and prepare a particular smear on the glass slide of this microbial culture. We prepare a smear first on this microbial culture. But one thing that needs to be mentioned here is that prior to preparation of this particular smear, this glass slide has to be made grease free. Otherwise, a uniform culture is not possible. Uniform smear rather is not possible. Formation of the uniform smear is not possible. So after the smear has been prepared, it is allowed to air dry, and then this particular smear is heat fixed. This step is very simple. Basically, you pass this smeared slide over a flame, from say a burner for say 2 to 3 times. The reason is that the microbes, they have a mucolipoidal layer on its surface which basically melts in presence of this heat, and the microbial cells get fixed on the glass slide. So why do you need to do this? Because in the next step, it's a staining step, you have liquid stains, destainers, decolorizing agents, and several steps of water washing. So if you do not fix the microbe on the glass slide, then the microbes or the particular cells might get washed away. So this is the step in brief of smear preparation. Next, we will be looking at the staining process. So what are stains basically? Stains are basically coloring agents. So these stains are basically coloring agents which are specifically used for coloring microorganisms. So they are basically the purest form of another coloring agent, that is the dye. So basically stains are the pure form of coloring agent or dyes which are used for staining or coloring microbes. So there are several different types of stains which are basically used for coloring microbes, but the most simplest stain is the crystal violet stain. This particular stain we use to just color a microbe and observe it under microscope. The staining process is called the simple staining. The step is very simple. Basically, you need to flood the heat fixed smear with crystal violet. Okay, you need to flood it with crystal violet. Keep it for 30 seconds and then wash the slide in running water. Okay, and then allow it to air dry. Okay, so this is the basic steps that are involved in simple staining. So In this way, the culture or microbial culture is stained. So, the next step or the final step of observing a microbe is the observation step, for which we need an instrument, which is the microscope. Now, microscope is an optical instrument which involves combination of lenses, and as we all know that lenses are used to create magnified image of objects. So the microbes which we are unable to see through our naked eye, when we see through lenses, we get a magnified image of the microbe, and the microbe can be, its structure can be revealed, or its shape or its size can be revealed. So basically the microscope, it involves two different types of lens system. One, it is the objective lens, and the other is the ocular lens. Though there is another lens that is present in the microscope, but that particular lens is not involved in image formation. That is called the condenser lens, which we can discuss later when we explain the parts of the microscope. So basically, in a microscope, there is a stage in which the smear that we had prepared and that we have stained, that particular prepared slide is placed, and through these two different lenses we observe that particular slide, that particular stained smear on the slide, and we get a magnified image of that particular microbe of which we had prepared we have prepared to observe. So basically, when we observe the microbes through these two lenses, we get a magnified image. So this is the way by which we can observe a particular microbe, and its shape, its size, its structures can be revealed. Hope you enjoyed this session, and please subscribe to our channel and click the bell icon to get more notifications about more videos from Microbiology Insights.